In this video, we will discuss about two different kinds of unfolded protein response. One is adaptive unfolded protein response, another one is proapoptotic unfolded protein response. In our previous videos, we have discussed mostly about adaptive unfolded protein response. So before going on to the main topic, let us first quickly revise what is this unfolded protein response. Unfolded protein response is usually produced in the endoplasmic reticulum, or not usually, is unfolded protein response is produced in the endoplasmic reticulum whenever the amount of unfolded protein increased beyond a normal beyond a normal or beyond a normal limit. So this will triggers the unfolded protein response from the ERs, and that will leads to this will trigger unfolded protein response because of the ER stress, as ER will be uh, more crowded with the unfolded proteins in case of more amount of misfolded proteins are available in the endoplasmic reticulum due to any reason. So there are two kind of conditions that can arise. One is that unfolded protein response can be reversed or it is controllable. And another condition in which unfolded protein response cannot be reversed and cell has to proceed towards programmed cell death or pro-apoptotic UPR. So let us just go through the adaptive unfolded protein response. It will leads to three kind of signaling. First one is PERK signaling, second one is IRE1 alpha, and third one is ATF6. So what will happen in the PERK signaling is that it will leads to this EIF2 alpha signaling pathway that will leads to phosphorylation of phosphorylation of this EIF2 alpha. After this PERK signaling gets activated, the EIF2 alpha gets phosphorylated. It will triggers the translation attenuation, and that will reduce the translation of proteins that are not necessary for controlling this unfolded protein response, and simultaneously it will upregulate the proteins that are involved in the unfolded protein response, or to control the unfolded protein response through this ATF4 transcription factor. So some of these are mentioned here, such as the ER protein folding, or we can say chaperones that are involved in protein folding will get increased, and the autophagy. So those proteins that are not being able to form their correct structure needs to be directed towards proteasomal degradation, so or through other ways such as autophagy. So and other things are also mentioned here. This thing will happen in the nucleus to trigger this upregulation of this kind of proteins. Next signaling pathway is the IRE1 alpha. The phosphorylation of IRE1 alpha will lead to splicing of this XPV1 mRNA, and after splicing, it will this will act as a transcription factor that will go to the nucleus to upregulate the proteins that are involved in the uh, unfolded protein response in some way, such as it will also upregulate the ER protein foldings and ERAD pathway. ERAD pathway is basically associated with the endoplasmic reticulum associated degradation. So those proteins that are not able to acquire their native structure even after several times in the calnexin and calreticulum cycle, they will be directed towards this ERAD through ubiquitination. And third pathway that will activate is the ATF6 pathway. So what will happen is that the this protein ATF6 proteins will move into the Golgi. It will get cleaved through the proteasome it will get cleaved to the with the help of proteolytic cleavage. It will get cleaved at proteolytic cleavage through some proteins, and after that a part of this ATF6 will move to the nucleus and act as a transcription factor to upregulate these proteins, such as the uh, proteins involved in protein folding, such as chaperones, and it will also upregulate ERADs. So all this common function of these three signaling pathways to upregulate the proteins that are involved in maintaining the proteins that are involved in providing the newly translocated proteins into their native structure, or we can say upregulation of chaperones and ERAD pathway. However, when the unfolded protein response crosses the threshold level, it will change its signaling slightly to trigger this proapoptotic UPR. So what will happen in this proapoptotic UPR is that each signaling pathway will act as will act differently. In the PERK pathway, what happens is that dephosphorylation of this PERK pathway will happen with the or will occur with the help of this PP1 and CREP1. So because of this, the what will happen, the efficiency of EIF2 alpha to function as it is functioning in the adaptive UPR will reduced, and it will move slightly, and it will change its pathway in a different way, such as it will start to producing a CHOP protein that is mainly associated with ERAT pathway. So it is the signature protein that can provide a signal that UPR that is observed within the cells is the proapoptotic UPR. So after this CHOP is been produced, so what will happen is that it will triggers this GADD34. It is related to DNA growth arrest and uh, DNA damage, and that will triggers this DR5, that receptors 5, and this that receptors 5 will finally, so with the help of BID, it will create mitochondrial stress, and this mitochondrial stress will triggers the apoptosis. So this is how the PERK pathway will leads to apoptosis. In IRE1 alpha, what happens is that this TRAF2 and JNK pathway will triggers this RIDD. It is related to degradation of mRNA, and this degradation of mRNA signaling pathway will triggers this BID and will cause mitochondrial stress that triggers apoptosis. This CHOP pathway will also cause any toxic uh, or we can say the CHOP pathway will activate other toxic pathways that will trigger the mitochondrial stress that leads to apoptosis. And in this case one more pathway that get triggers, that is IP3R, that will leads to increase in the amount of calcium, increase in the amount of calcium in the increase in the amount of calcium in the cytosols. So that will cause uh, stress on the mitochondria, that will also cause apoptosis, or we can say this increase in the Ca2 will leads to inflammatory response. So it will initiate this caspase 1 and interleukin 1 beta that will leads to sterile inflammation, and finally this interleukin 1 beta will finally end up triggering this apoptosis pathway. So this is how the proapoptotic UPR triggers towards apoptosis pathway and how it is different from adaptive UPR. So I hope you have understood some idea about this proapoptotic and adaptive UPR. If you need to understand this thing in details, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.